Hello, good morning, everyone. Hope you guys had a good weekend. Let's get started with today's tutorial. So we will be working on lecture 3, and when more people come, then we can take up some of the questions from the quiz. But let's start with this study guide. Okay. So lecture 3 is about cells, and here's the first question. State the 4 principles of the cell theory. So let's write that down. So the first one states that. First one states that every living organism consists of one or more cells. Okay, so if you think about like bacterium, right? That's like one cell. Okay, if you think about like a cat, right? Cat goes to many cells. Okay. Humans have like trillions and trillions of cells, right? So every living organism is going to have one or more cell. That is the first one. The next one. A cell is the smallest unit of life. Okay. Individually alive as part of a multicellular organism. Okay. Give me a second. Someone's emailing me about the link. Give me a second. Okay, so that's the second part of the cell theory. And then the next part of the cell theory is that all cells come from the division of pre-existing cell. Okay, this is as you will learn later in the course. This is called mitosis, right, which basically means cell division. Okay, cell division. And then the last part basically states that cells contain genetic information that can be, sorry, it's like always running out of space, that can be passed on during cell division or mitosis. Okay, so there's this ability to pass on genetic material, to copy all of its genetic material and essentially clone itself. So these are the 4 principles of cell theory. Okay, next we're going to fill in the blanks with terms that correctly describe the organization of life from the smallest to the largest. So we start with atoms, and we learn about, different types of elements last class, and these elements can come together to form molecules, right? Okay, so like small molecules like H2O, maybe like C6H12O6, like a sugar molecule. So those are small molecules. Okay, but then we also learned last class that there are these big molecules, right, with hundreds and thousands of atoms in it, and we call them macromolecules, right? Macromolecules. Okay, and then these macromolecules are basically the building blocks for other structures, okay, such as organelles. Okay, so organelles are like tiny organs, tiny structures that are found throughout the cell, and of course that's what this lesson is about. They all have various dedicated functions, which will be essential for the normal functioning of the cell. And then the organelles would make up cells. Okay. And then many different cell types can come together to form tissues. Okay, so anybody have an example of a tissue for me? Anybody? Give me an example of tissue. Yes, scar tissue is an interesting one. Yeah, that could happen. But let's talk about like in a normal healthy state. What kind of tissue do we have? Okay, so muscle. Yeah, muscle could be a type of tissue. Okay, anything else? Soft tissue. Yeah. Fat tissue. That's right. So a bunch of different types of tissues, right? Connective tissue, right? So these are essentially a collection of specialized cells that would perform a specific function. So in fat, we have these adipocytes, okay, fat cells. And their function is to store fat as nutrients, okay. Nervous tissue, that is also correct. That's what Allison said. So yeah, those are a bunch of tissues. Now we can take a collection of tissue and form an organ, right? An organ. So an example of an organ would be something like the heart, right? Liver. Right? So these organs have a bunch of different types of tissues. So the heart, for example, they have connective tissue there, right? There is some muscle for contraction. There are some adipose tissue, right, that insulates the heart. Okay? And then you have multiple organs in an organ system. Organ system. So does anybody know any organ system? Any example of an organ system? Yeah, digestive system. And what might be in the digestive system? What kind of organ would be in the digestive system? Yeah, there's the stomach, right? There's the liver, gallbladder, intestines, a bunch of organs within the digestive system. Yeah, esophagus. Very good. Okay, so this is the organization, yeah, of smallest to largest. Yeah, respiratory system, that's another one. Very good. Okay, next one. List 3 common structures found in all cells. Okay, so cells, they look different. Okay, if you look at bacteria cell, it looks very different than a human cell, which looks very different from plant cells, and they are kind of unique in their own way. However, there are 3 common structures that we are going to be able to find in all cells. So the first one is cell membrane. Cell membrane. So another word for cell membrane, right? Like a fancy word, it's plasma membrane. They mean the exact same thing. Okay, exact same thing. So this is the barrier that separates the living world on the inside of the cell from its non-living environment. Okay? The barrier that controls what goes in and what comes out of the cell. Does anybody remember the major component of cell membrane? We talked about that last class. What is the major component? Yes, phospholipid bilayer. That is correct. Okay, so the next part is cytoplasm. Cytoplasm. Okay, that's the fluid, kind of like a gel-like substance that is inside the cell membrane, where the organelles are going to be floating around in. Okay, it's not alive. It's just a gel-like fluid. Okay, and finally, they all have genetic material. Okay, so like DNA, right? We talk about that. Okay, so these are common things that are found in all cells. Okay, so basically, like you can think of like a bag here. That's the cell membrane, and then you will have the cytoplasm. So like cell membrane. Call that 1. 2 is cytoplasm. That's the fluid inside, and then you will have some genetic material. Okay, number 3. The genetic material might or might not be in a concentrated area. Okay, depending on whether it's a prokaryotic cell or a eukaryotic cell. Okay, so let's take a look at the difference between prokaryotic cells and eukaryotic cells. So another name for prokaryotic cells is prokaryotes, right? Prokaryotes. Okay, and then this another name is just eukaryotes. All right. So prokaryotes, right? P for primitive. Okay? These are cells that are rather simple. Okay, they are not too complicated. So in terms of size, they are going to be smaller. Smaller. Okay. Simple structure. There's not a lot of moving parts, so to speak, right? It's very, very simple. And the location of genetic material is kind of in the central area of the cell. Okay, not compartmentalized. Okay, there's no separate structure to keep them in a place. Okay, so like if you think of a bacterium, here's a bacterium. Bacterium is the singular for bacteria, right? If you have more than one bacteria, then it's called bacteria. So you would have the genetic material in the center. Okay, so genetic material. And actually, bacteria, they have these little circular DNA as well. It's just going to be floating around. Anybody know what they are called? Just wondering. Circular DNA, anybody know what they're called? Good try. These are circular DNA called plasmid. Okay. Circular DNA. Okay. If you don't know that, that's okay. We will have a whole lecture on that later on in the course. Okay, but these are special DNA that gives the bacteria like special abilities. Okay. Anybody have heard of like superbug? Superbug. You may have heard of that on the news. Yeah, superbug. No superbug. Superbugs are these bacteria that are very resistant to antibiotics, and you know, if you catch them, it's really hard to cure. Okay, and sometimes we don't have drugs for them, and you would die from it. Okay, so these plasmids, one of the things that it could do is it turns them into superbugs. Okay, so more on that later. Okay, number of organelles, few. Okay, not a lot. An example would be, like I said, bacteria. Right? Bacteria, such as, anybody have know any bacteria? You read it on the news, you read it from a book, you learn it from before. Any example of bacteria? E. coli, yes, that's probably the most famous one. Many people know E. coli. Not many people know what the E stands for. I know what it stands for, but I cannot pronounce it, so I'm not gonna try. Another one? Anybody have another one? Coccus. Yeah, coccus is actually a shape, right? Coccus means balls. So there's streptococcus, for example, right? That causes like strep throat. Okay. So yeah, S. aureus, for example, that's a streptococcus. Okay. Yeah, Allison's right. The E. coli, that's what it stands for, right? I cannot pronounce it. It's like Escherichia or something like that. But you got that right. Okay, which is what they all have very complicated names. Okay, great. What one more? Maybe one more. Just to see, right? Another common one that you hear on the news. They recall the salads all the time, right? Because you know, of this contamination. They recall you know, Delhi because of contaminations of Salmonella, right? Salmonella. Okay. Salmonella is naturally occurring on some reptile. So they say, you know, if you handle like a snake or a turtle or whatever, like you gotta wash your hands because you might get some salmonella on you. Anyways, these are examples of prokaryotic cells. And just because they're smaller, they're simpler, doesn't mean they are they don't pack a punch, right? As you know, if you get infected by bacteria, it could sometimes kill you. Okay, flesh-eating bacteria, right? You might have heard of that before. Okay, eukaryotes, that's like us, right? They are larger. Okay, the structure is complex. Okay, genetic material is within the nucleus. Okay, and the number of organelles, many. Okay, so we learn a bunch in this class, right? The ribosome, the mitochondria. We'll talk about all that. Example: human cells, plant cells, right? Fungus, right? Fungal cells, right? All these things are eukaryotic cells. Okay. Now I just want to put a like a little asterisk here, right? Eukaryotic cells, although they're supposed to have a nucleus, but there are exceptions, right? So some human cells don't have a nucleus. Okay. Anybody know any example of a human cells that do not have a nucleus in it? Yes, Tyler said RBC, that's correct. Red blood cells, right? Another example is platelets. Right? These things helps you clot the blood. They do not have a nucleus. So there are exceptions, right? Like in science, there is not always like binary, right? Not always this or that. There are some exceptions. There are some gray area. Okay. All right. Let's see if we can do some polls. Just to see where people are. Let's do this one. All right. Where's the poll button? Go ahead. Okay, very good. Most of you have got it. Again, please do participate in the poll. Right? The following is a concept of cell theory. A cell is a basic structure and functional unit of living things. That's correct. Okay, let's try another one. Relaunch the poll. Okay, all of the following present in all cells except, okay, so we literally just did this question, right? List 3 common structures found in all cells: cell membrane, cytoplasm, genetic material. So all we have to do is look, cytoplasm, plasma membrane, genetic material, right? So the one that is not found in all cells would be nucleus. Okay, nucleus. And that's how I kind of felt when I look at the results of the quiz. Some of the questions are the exact same question that we did in class, right, from the tutorial, and people still didn't get it right. So perhaps you didn't come to the tutorial, or perhaps you didn't revisit some of those poll questions that we did. But hopefully, right? Hopefully you would know to do that now, okay? Because that's what these polls are for. It's supposed to help you prepare for the quiz. Having said that, the class average is pretty good. 73 is pretty decent, and we will take up some of those questions maybe in a bit. But let's keep going for now. Label the different parts of a mitochondrion. Okay? The mitochondrion is the powerhouse of the cell. It is the organelle that makes ATP, right, in the process of cellular respiration. Again, we will have a whole lecture on that later on, lecture 5. But now let's just take a look at the different parts of our mitochondria. Okay, so it is a double layer membrane. It has a double membrane. Sorry, double membrane. The outer one here in green, that's right here, outer membrane. Okay, and then there is also the inner membrane. The inner membrane. Let's see if I can highlight that for you. Okay, so this inner membrane folds up into these accordion-like structures, right? Finger-like projections. That's what some textbook calls it. Okay, so that is the inner membrane. Inner membrane, right? It folds up, folds into finger-like projections called cristae. Okay, and that actually help increase the surface area for reaction to take place. Okay, we have another one right here. This is pointing at the space between the outer membrane and the inner membrane. Okay, so like let me draw it a little bit nicer here. This is the outer membrane. This is the inner membrane. Okay, and then the space between it, that is called the intermembrane space. Intermembrane space. Okay, and finally, the fluid on the very, very inside. That is the matrix. the matrix. Okay. So, fun fact. Did you know mitochondria have its own DNA, which encodes for some of its protein? Okay. Now, because it's DNA, it must be inherited from the previous generation, right? So who gives us our mitochondrial DNA? Let's do a poll and see if you know. Okay. Who gives us our DNA, our mitochondrial DNA? Is it dad? Choose A. Is it mom? Or is it both? Okay. Who do we get our mitochondrial DNA from? All right. It's from the mom. Okay. All the mitochondrial DNA are exclusively from the mom. Okay. If you think about what happens is there is the egg, right, with all the mitochondria in it, and then here's the sperm. Okay. When the sperm comes, the lucky one that gets to put in the DNA into the egg, all right, the one that forms us essentially, it only injects the DNA. Okay, so all the organelles here, the mitochondria will continue to divide to grow, and that in turns gives us all our mitochondrial DNA. Okay, so it's kind of neat that way. Okay, let's move on to the next part. Here is describe the functions of each component of the endomembrane system. What is the endomembrane system? Okay, endomembrane system is basically a series of membrane inside the cell. Okay. And all these membranes of the endomembrane system, they have interrelated functions. Okay, they're all related to each other. So on the next quiz number 2, which is 3 and 4, there might be some diagrams. Okay, I might show you a picture and ask you what it is. So make sure you're going to be able to identify them. Okay, so the first one here, this one, let me just zoom in for you. This one is the rough endoplasmic reticulum. You see all these dots that are on it, right? Actually black dots on the diagram. So all these dots are the ribosomes, right? And that gives it like a rough appearance. That's why this part is called the rough ER, rough endoplasmic reticulum. Okay. Rough ER or RER. Okay. So let's put in some description here. Okay, let me try to fit all this stuff in. Okay, so description, I'm just going to do that in pink maybe. Okay, it's part of the endomembrane system. You know what? They are all part of the endomembrane system. Maybe I don't have to write that. like everything in this table is part of the endomembrane system, okay? And one of the thing about rough endoplasmic reticulum is that it is continuous with the nuclear envelope. Okay, so if you're a little bit confused about what that means, let me quickly show you. Okay, I'll come back to this slide. So this is the nucleus, right? Nucleus. That's where you will find the DNA, the genetic material. And then this nucleus is actually surrounded by its own nuclear membrane. Okay, we call that the nuclear envelope. Okay, it's kind of like the orange peel of an orange, if you would. Okay, it surrounds it, and then it will continue to extend into the cytoplasm. Okay, you don't have to draw this. Okay, and then there will be these ribosomes on them. Right, so this here, that is the rough ER. Okay, so it is continuous with the nuclear envelope. Okay, that's what it means. Okay, so back to here. It is covered with ribosome. Okay, covered with ribosomes. And ribosome is what makes the protein, right? Okay, so the functions, what is the function of the rough ER? Okay, so basically the ribosomes synthesize proteins directly into the inside of the rough ER. Okay. The proteins will be packaged into a vesicle by the rough ER. Okay. All this is in the PowerPoint, of course, which is, writing it down, summarizing it. Okay. So if you think about like this is the rough ER, right? For example, rough ER. Okay, and then here is my ribosome sitting on it. So ribosome, maybe ribosome right here, ribosome. Okay, and what happens is the ribosome will make a protein, right? The polypeptide that we learned last class, the amino acid. So the protein will be synthesized directly inside the rough ER, and then this thing will fold up into its, three-dimensional shape. It will migrate towards the edge, and then a bubble will kind of butt off from the edge with the proteins in it, right? Okay. So the inside of the rough ER is hollow, right? That's where the protein is going to go. And it's going to flow towards the edge. It's going to fold up in its final shape. And this part's going to pinch off into a vesicle. Okay. So to summarize in one very simple sentence, the rough ER helps make proteins and package them, right? Along with ribosomes. Any question? Okay. Next part is the smooth ER. It is still continuous with the rough ER. Okay, but these are smooth because there are no ribosomes on them. So smooth endoplasmic reticulum. SER. Okay, let's talk about this stuff. It's still connected to it. It's further out from the nucleus. Okay. And let's go through the description first. Okay, continuous with rough ER, but not directly touching the nuclear envelope anymore, right? Like we're really, really far out. And they are not covered not covered by ribosome. So it appears to be smooth under the microscope. Okay, what does it do? What does it do? Okay, it is important for synthesis of lipid. Okay, they make lipids. Okay, so give me some examples of lipids that we're gonna need to make. Something that we learned last class. Anybody? Hormones, that's right. What kind of hormones? Give me an example of a hormone that we would make from the smooth ER. Exactly, right? Estrogen. Okay, so this would be like example like steroid hormone. Okay, so you would expect, one would expect a cell from one would expect an ovary cell to have lots of smooth ER, right? Okay, ovary, that's where you make the sex hormones, right, for a female. And for male, it is the testicle, of course. Now, the smooth ER also has another role other than making lipids, right? They are also involved in the breakdown of lipids. Okay, not only do you make them, you also break them down. You can break them down into its components, and then maybe you can use those components to make the lipids that you need. And detoxify drugs. Okay, so for example, in which organ? What organ is responsible for detoxifying drugs? The liver, that's right. Very good. When I say drugs, I don't mean like, illegal drugs necessarily, okay? If you take like Tylenol, right? It's gonna get metabolized in the liver. Ever see, like, read the label warning on the Tylenol? It says like, do not take more than whatever, right? Like may cause irreversible damage to the liver, right? It actually says that. You can read that. Okay, so like a lot of drugs that we take, antibiotics, all that kind of stuff, the detoxification, the breakdown, the metabolism of it happens in the smooth ER, in the liver. Okay, any question about the smooth ER and the rough ER, how they're related, what's going on? Okay, so bottom line is this, right? Listen carefully. Okay, write this down if you need to. If I say, there is a cell and the function is to make a lot of proteins, do you expect it to have a lot of rough ER or smooth ER? Okay, so if it's involved in protein production, lots of protein production, then you would expect it to have a lot of rough ER. If it's involved in lipid production, then you're gonna expect it to have a lot of smooth ER. Okay, smooth ER. Get it? Great. All right, next part. Next part, we have the this thing. Anybody know what that is? What is that I'm looking at? That is the Golgi body or the Golgi apparatus. That is correct. Okay, so Golgi apparatus. Okay, or body. Doesn't really matter. Okay, so Golgi body works like this. Okay. Well, let me give you the description of the structure first. Structure is that it consists of layers of membrane stacked together, if you would. Stack together. Although it's not like literally touching, but they're like really close to each other. Okay, and the function is that it will sort, oops, wrong color. It will sort, modify, and repackage proteins. Okay. And this is basically how it works. Oops. All right, so we're gonna have a, you know, 1 stack, 2 stacks, 3 stacks, 4 stacks, whatever, it doesn't matter. And what happens is we're gonna have a vesicle that's gonna be coming from, say, the rough ER. Okay, inside this vesicle we have a little bit of a protein. Okay, now this protein might or might not be completed. Okay, there might still need some things to be added on. I think I told you earlier, maybe I haven't, I don't remember, that sometimes proteins, you need to have some sugar on it. Okay, then it turns it into like a sugary protein. We call that glycoprotein. Sometimes you might have to add some lipids to it, right? So it's going to be like a lipoprotein. And all that modification, okay, some of it takes place in the Golgi apparatus. So this thing's going to come and it's going to fuse with the first layer, okay, and then the protein is going to get inside, and then it might get modified. Okay, once it gets modified, then it will get split out in a new vesicle. Okay, so this is the same protein, but now maybe they added some stuff to it. I don't know what they add to it, doesn't really matter. And then this is going to get put towards the next layer, and then the next layer, and sometimes they will go back and forth and gets like sorted out and gets remodified several times. It's kind of like an assembly line, if you would. Okay, so you know, first person do something to it, pass it to the next, and then pass it to the next, and then you might have some QC people, quality control. They look at it, it's like, this is not good yet. And they send it back, and then they modify it. So there's this back and forth going on in the Golgi body until finally it's done. Then you would have the final version of this protein, right? Okay, like maybe it has the red dots on it, and then whatever, some green dots. Okay, so this is the function of the Golgi body. Okay, now where does this go? Right? Where will this protein go? Okay, this vesicle. Okay, so we should talk about that. Okay, the potential possible destination of the vesicle. Okay, where is it going to go? Well, number one, okay, it might become a lysosome, which is what we're going to talk about next. Okay, so this guy here, that's lysosome, right? That's responsible for like digesting stuff, which we can talk about in a bit. What's another possible destination for this guy? Anyone? Please tell me. Where is it gonna go? I'm just looking at the chat, see if anybody can tell me where else is it gonna go. Not necessarily gonna go back to the nucleus. Anyone else? Outside of the cell, that's right. So in the proteins, the contents, we'll say, okay, the contents can be released out of the cell. Okay, so maybe this is insulin and you need it now, right? Because you just had a meal. So this is going to go all the way up. Let's say this is the cell membrane, right? Watch, this is going to go, and then it's gonna we call it like the kiss and fuse. It's gonna kiss the cell membrane, okay, and then again this is the protein inside, and then what's gonna happen is this will fuse with the cell membrane, right, and then all the content is gonna be released out of the cell. Do you understand that? I know this is not the best animation. You should watch some of the videos that I post. It's probably much easier that way. And then yes, Tina says store for later. Yeah, so this could, maybe you don't need to release the insulin now, right? Maybe you want to store it, so it would just, you know, stay in the cytoplasm until cannot spell until it's needed. Okay, and number 4, it's also possible that it will embed into the membrane. Okay, so some of the proteins, they are actually going to stay on the membrane and perform various functions that we will talk about later. All right, so these are the possible destination for our vesicle, for the proteins. Okay. All right. So why did I tell you that? Well, this question was done poorly last semester. So hopefully you will know the answer after this brief discussion I had with you. So go ahead. Choose the right answer, people. You can do it. Okay, has been a minute. I only got 8 response. I'm gonna wait a little longer. The answer should be quite obvious based on what we just discussed. All right, I'll end the poll. Now, I must say I'm surprised. I literally just talked about this like a minute ago. How come a lot of people still choose other things, right? The correct answer is like this one, right? Like except, right? Where will you not find it, right? So these are all the locations that we've talked about. Okay, so I want to know, right? Like for those of you who chose something else, like if you don't mind telling me, like it's something confusing, I cannot help you if I don't understand what's confusing you, right? Like, or maybe you just missed that little segment that we had earlier. Like for example, why a lot of people chose like embedded in cell membrane. How come? Right? I told you, right? It's possible that it get embedded in the cell membrane, right? So is there a confusion that I should know about? No confusion. All right, well, hopefully there's no confusion when the test comes. All right. Okay, next one then. We'll move on. Lysosome. Right, this is the lysosome. Okay, so the description, it is going to be, again, part of the endomembrane system. And it's a small sphere, small vesicle, sphere that contains various digestive enzymes. Okay, and in terms of functions, what it does is it can break down break down macromolecule. Okay. And things the cell engulf, right? Can also trigger programmed cell death. Okay, we call this apoptosis. Okay, deliberate cell death. Okay. So macromolecules, if you eat, you know, your carbohydrates, starch, whatever, you need to break it down into smaller parts, right? Or you need to break down the proteins. That's going to happen in the some of it is going to happen in the lysosome. Sometimes the cell can eat things up. Okay, so for those of you who do not know, like a white blood cell. Okay, let's say this is the white blood cell. White blood cells are your defensive cells, and they seize like a bacterium. So what happens is the white cell will move towards it, okay, and then it will basically like eat it up like that. Okay, so afterwards, the bacterium will be surrounded by a vesicle, okay, like this, and the vesicle will fuse with the lysosome, and then the digestive enzyme will break down the bacteria. Okay. Sometimes we want a cell to die. Either it's too old, right? It's not functioning. We want it to die on purpose. So it will release all its content. The lysosome will release all of its content, causing the cell to die. Okay, we call this apoptosis. In the lecture, I mentioned like during development, right? Our hands used to have webs, right, in them during development, and we want the webs to die away, right? The cells in the web, so that our fingers is going to be separated. So that is called apoptosis. Okay, now if the lysosome the lysosome has a lot of digestive enzymes in it. If some of those enzymes are not functioning, okay, because of say like a genetic problem, okay, there is a mutation that causes the enzymes to misfold, because enzymes, as we learned, are proteins, right? And the instruction to make those proteins are encoded within the DNA. So if you mutate a DNA, that could cause the enzymes to be missing or not working. So that could lead to a bunch of lysosomal diseases, right? So in the lecture, I mentioned a few examples. Anybody remember what they are? Just want to see. The question is, what kind of lysosomal disease could happen if the enzymes are missing? Okay. Genus Progeria. Progeria is actually related to the lemon A, which is one of the cytoskeleton. We'll get to that later. Yeah, Pompey's disease. That's correct. Okay, so for those of you who don't remember, Pompey's disease is because one of the enzymes that's needed to break down glycogen is missing. So you have glycogen accumulation. Okay, in muscle. Okay, especially the heart, and those kind of accumulation could be toxic to the cell, interfere with the function, and lead to a bunch of problems. Another one that we talk about, exactly as Alison mentioned, is the Tay-Sachs disease. Okay, so in Tay-Sachs disease, it is the accumulation of lipids, okay, in brain, because you're not able to break them down. All right. So depending which enzyme is mutated, the kind of stuff that gets accumulated is going to cause different outcomes, different diseases. All right. Okay, moving on. List 3 organelles or structures that are unique to plant cells. Okay. You know, I actually meant to change this questions because it's a little bit misleading. Instead of saying that are unique in plant cells, let's just say that are found in plant cells, but not animal cells. Okay, because some of the stuff is found in plant as well as things like you know, fungus and stuff like that. So I just want to compare like plants and animals in this case. Okay, so first one, right? Anybody, please please tell me something that you will find in plants but not animal. Anything at all? You guys have been really quiet. Anybody want to unmute? Or, oh, typing is fine, I guess. Yes, chloroplast. Let's write that down. Chloroplast. Okay, so what is the function of chloroplast? It contains chlorophyll that enables photosynthesis, right? Okay, so chloroplast is found in plants, but not animal cells. However, right, you can find chloroplast in other types of cells, right? Protists, for example, right? Some protists, they have chlorophyll in it. Okay, so what are protists? I don't know if you guys know this, but like there are different kingdoms, right? In terms of living things, there is the animal kingdoms, which includes like humans, cats, dogs, like all the animals that you can think of. There's the plant kingdom, right? And then there is the bacterial kingdom. And then there is also fungus and protista, right? Or protists. Those are separate kingdoms. So these are like, you know, the best example that you can relate to probably is amoeba. Amoeba is an example of a protist. These things tend to live in like pond water, for example, in legs, right? So some of the protists, they would have chlorophyll. My point being, the chloroplast is found in plants, but not animals, but they are not necessarily unique or exclusive to plant cells, and that's important to note. And then there is a bunch of other stuff people wrote on the chat, which is excellent. Next one is cell wall. Okay, so cell wall, it's made up of cellulose. We learned that last class, and it protects the cell from bursting. Okay, and provide like structural support. And as you will learn next class, you know, you probably know this based on common sense. Plants loves water, right? So like you water them, it helps them stand up. But the thing about cell is if you put them in an environment that has a lot of water, they tend to swell up and burst. By having a cell wall, it kind of protects it from bursting. And again, right, we don't have cell wall, animals don't have cell wall, but other things could have cell wall, right? Like a fungus, right? They could have a cell wall. So by no means it is unique to plant, which is why this word is shouldn't be there. I meant to change it, but I keep on forgetting. Okay, and then the last one, some of you say like vacuole. Yeah, so they have like what's called like a central vacuole. It's like this big structure in the center of the cell. Like if this is the plant cell, there is going to be like a vacuole that takes up like 70 to 80% of its space, like a super big swimming pool in the middle. And what it does is it will stores water and nutrient. Okay, and in some cases can even store pigments. Pigments are like colors, right? That gives the petals like its beautiful colors, or poison. Okay, if the plant is poisonous. Okay, so there you have it. Those are the 3 organelle structures that are in plants but not animal cells. All right, so that was an hour. Why don't we take a quick break? 5 minutes. We'll come back at 9:35, and we will resume shortly. See you in a bit. And we are back for more biology. All right, moving on to the next page. Complete the following chart. Okay, so these things are no longer part of the endomembrane system. Okay, they're just that. Okay, so here we have this big purple thing. That is, of course, the nucleus. Okay, they are found in eukaryotic cells such as human cells, animal cells, plant cells, fungus, but not in bacterial cells. Okay, so a description of it. Let's write down. Okay, so found in eukaryotes, with some exceptions that we mentioned earlier. It is surrounded by nuclear envelope, which is just a membrane, right? Okay, and this nuclear envelope, as I mentioned earlier, is continuous with the rough ER. Okay. On the nuclear envelope, there are many nuclear pores, okay, which allows things to go in and out of the nucleus. Many nuclear pores on the nuclear envelope, okay, which allows, again, things to go in and out of the nucleus. And you can see all these nuclear pores right here. Okay. So what is the function? Well, the function is that it contains DNA, right? Okay. As well as there is another part inside the nucleus, which is this central part here. Okay, this part that is called the nucleolus. So it contains nucleolus, which makes ribosomes. Okay, so let me write that down for you. This is nucleolus. All right, I want you to think of this as like maybe like a peach. Okay, so the peach, the skin, the fuzzy skin, that is the nuclear envelope. The meat, the meat of the fruit, that would be the location where you will find the DNA. Okay, and then the seed in the middle, that's the nucleolus. Okay, so the nucleolus is going to make ribosomes, and then you will have all these genetic material here, the DNA. Okay, now these DNA are actually packaged. Okay, the DNA is combined with proteins, something called histone proteins, to form chromatin. Okay, so this chromatin is basically like a condensed version of the DNA. Okay, so you know, your DNA would be like just the ACTG, but that's like too long. It will never fit into the nucleus. So you would add a little bit of the protein, and so it's gonna coil up. Okay, so that's gonna fit into the DNA, the nucleus, chromatin. And then when you are ready to divide, when the cell is ready to undergo mitosis, it will continue to condense further, and then it will form like the structure that we all are familiar with, like what we see in like textbook diagrams and stuff like that. This is the chromosome. Okay, so chromatin and chromosome, they are essentially the same thing, but the chromosome is much more condensed. It's easier to move them around without breaking them when it's really condensed. Okay, so this chromatin, which is DNA plus protein, it's found in the nucleus. Okay, so that's one of the functions of nucleus, right? It contains the DNA, it protects them. That's basically That's basically what it is. Any question about any of these? All right, next one. These little dots. Okay, they're barely visible on a regular diagram, but if you zoom in, it actually has two parts to it. We call these the ribosomes. Ribosomes. Okay, let's go describe them. They might exist freely in the cytoplasm, just floating around. Okay, or be associated with the endoplasmic reticulum, right? Which would then turn the endoplasmic reticulum into like a rough endoplasmic reticulum. It actually has two subunits. Okay, the large subunit and the small subunit, and it's made up of RNA and protein. Okay, that's what makes up the ribosome. So there's a little bit of RNA in it. There's a little bit of proteins in it. And then the function is protein synthesis, right? It makes proteins. If it's freely flowing in the cytoplasm, then it would just make the protein, and then the protein would, would not be packaged in a vesicle. If you need it to be packaged in a vesicle, you would have to make it in the rough ER. Okay, so now is a good time to talk about the central dogma of biology. Okay, dogma is like the theme, essential theme of biology. So the flow of information in biology typically goes like this. You start with DNA, which is in the nucleus. Okay, you're gonna make a copy of the DNA in the form of RNA. Okay, in the form of RNA. So that is going to also be in the nucleus, but then afterwards it will exit into the cytoplasm. Okay, so move from nucleus to cytoplasm. And in order to do that, it's going to go through those little nuclear pores, right? Okay, so this process of copying part of the DNA in the form of RNA, we call that transcription. Okay, and that happens in the nucleus. Okay, think of the DNA as this encyclopedia in the library. Okay, but they won't let you take out the whole book. Okay, because it's very special or something. But then you want a few pages to do your research assignment on. So what do? You photocopy the few pages that you need, okay? And then you take it home with you. Okay? So this transcription allows you to copy part of the DNA that we need. And what do we need it for? Well, we're going to use this RNA, this small copy of the DNA, we're going to use it to make proteins. Okay. And this process of using the RNA to make the proteins, we call that translation. Translation. Okay. And that happens in the cytoplasm. Okay. In the cytoplasm. Okay. So how do you do that? Okay. So there is your RNA. This is your RNA. It's actually called messenger RNA. mRNA, right? That's what the vaccines from Pfizer, Moderna, that's what they're made of, right? The mRNA vaccine. So we naturally make mRNA in the body. And then what happens is you're going to have the ribosome. The ribosome will have the large subunit and the small subunit. Large subunit, small subunit. Many people call this the hamburger and the spaghetti model. Okay, so it clams on like a burger, it clams onto this RNA, and then it will read the instruction on the RNA to make the protein. Okay, so the RNA will tell the ribosome what amino acid to link together. So you would, take one amino acid, two amino acid, and then you will keep on linking them together like this. Okay, so this is the polypeptide. Okay, the protein. Okay, and then what's going to happen to this protein? Well, again, it could get packaged and then get sent to another part of the cell. It could be released from the cell, and so on and so forth, right? So just a little bit of like simple biology with the vaccine and stuff, right? So the mRNA vaccine, right? How does it work? Well, basically, the vaccine contains mRNA, which is this type of RNA, and this RNA allows you to make those spike proteins, which is the proteins that are on the coronavirus, right? Okay, the COVID virus. So you inject this into your arm, and then what happens is your white cells, one of your white blood cells, okay, will come along, and then it will pick up those mRNA. Okay, so once the mRNA goes into our white blood cells, right, the ribosome will come, and then it will make all these spike proteins. Okay, so there is a signal on the spike proteins that tells the cell, once you make them, you put it on the surface of your cell. Okay, so they put all these spike proteins on the surface of the white cell, and that allows your immune system to get activated. So then you would start to make antibodies that would target these spike proteins. Now, the spike proteins are harmless. They don't cause you to actually have COVID. Okay, it's just part of a structure on the virus. So now that you make all these antibodies, right? So when the real virus comes, these antibodies will be able to attack the virus and protect you that way. So essentially, that's how the messenger RNA vaccine works. Okay. All right, so let's go back to here. Okay, so essentially, what makes the polypeptide, the protein? The ribosomes. Okay, next one, we have this mitochondria. Mitochondria. Too thick. Give me a second. Mitochondria. Okay. So what does mitochondria do? Well, mitochondria, the structure we already talked about, so I'm not going to rewrite them. Okay. See question. What was it? Question 5, right? For structure. So what does it do? It is the site of cellular respiration. Okay, so that's where you will have C6H12O6 plus O2 producing carbon dioxide and water, and more importantly, ATP, right? ATP. So for, you probably would learn how to balance equation in chemistry, but for every glucose, we're gonna make up to 36 ATPs. Okay, so that's where it takes place. Next, do you know why some people get sick and some not after the vaccine? Yeah, we actually gonna learn that like next semester, right? But like long story short, when you say get sick, you don't mean like actually catching the disease, right? Because it's impossible to get the disease from the vaccine itself. Okay. When you say sick, I assume you mean like getting a fever or like getting the chills, right? Some of you might have got the vaccine and then, like for example, I got the vaccine, the first dose, I actually had a fever for 1 day. Second dose, nothing, no symptoms at all. And then the third dose, I got like muscle aches, right? Like, so those are not like sick necessarily, a sickness, right? Those are just a sign that your immune system is reacting, because even though the vaccine doesn't give you anything that would cause the actual illness, it is nevertheless a foreign substance that's being injected into the body. And anytime there's a foreign substance that is introduced into your system, your immune system would react. And some people would have stronger reactions, others not so much, right? So for those who have a stronger reaction, would more likely experience, the symptoms, right? The fever, the chills, and so on and so forth. Okay, and there is a misconception, right, out there, and I hear my relatives say this. Oh, I didn't really feel anything after I injected. I got the vaccine. It must mean that it's not working. Not necessarily. It's not really a correlation between, the severity of the side effects versus how effective it is, right? It doesn't actually correlate. It's just based on, how your immune system works. Okay, it doesn't necessarily produce any symptoms in some cases. Like my daughter, she got both shots, and nothing happened to her. There's no side effects at all. I wouldn't say it's an indication of whether it works or not. Okay. All right. Good questions. Now let's go back to this thing. We have the centriole. The centriole. Okay, so what are these things? Well, they usually exist as a pair. Okay, and typically arranged perpendicularly with each other. Okay. What does it do? It produces spindle fibers during mitosis, cell division, which would guide the movements of chromosomes. Okay. Pretty simple. Okay. What are these things? Help me out here. What is that stuff? Yeah, those are the cilia. Okay. So this is a hair-like structure that usually covers the entire cell. Okay. So what does it do? Enable movements. Okay. Or to move things around. Okay. The two here sound very similar, but let me explain. Okay. So like I said earlier, right, there are these like protists, right? These are like single cell organism that lives in a pond. And, if you remember high school biology, if you took that, there is one that looks like this, right? The paramecium, with the mouth here. There's like a special vacuum here, vacuole, and they are covered with cilia. Okay, so this is a paramecium. You don't really have to know the name. I just feel like writing it out. So all these cilia helps it move around in the pond, right? Okay. So let's talk about humans now. Where in the body will we find cilia? Anybody? Anybody remember from the lecture? Good try, not the esophagus. Esophagus has no cilia. You just squeeze the food down as if like you are squeezing a tennis ball through a stocking. Okay. Yeah, not necessarily the ovaries, but like close enough. There's the ovary. There is the fallopian tube, okay, which guides the egg, right? And then this is the uterus, right? So when the egg comes out from the ovary, right, there will be like cilia and the fallopian tube that guides the legs, the eggs towards the uterus. Okay, so it moves things around, like it moves the eggs towards a particular destination, right? And yes, trachea, right? There is trachea is the windpipe, right? There are cilia that would help move the trapped particles in your airway and move it out of your system. Not so much in the lungs though, okay? But like in the trachea, in your bronchi, right? And the those are the locations, okay? The upper part of your lower respiratory tract, if that makes sense. Okay, this one is flagellum. Flagellum. Okay, so usually just one. Right? Usually just one or two per cell. Okay, and then the function is movement. Okay, movement. Right, so sperm only has one tail. If a sperm has two tails, it will not be able to swim. It just go around in circles. You can search up on YouTube like sperm with two tails, and you will find interesting videos that shows the sperm going around in circle. But there are some protists that has like two tails. Okay, two flagella, okay, LLA, and then they can move around like that. Okay? This guy is called Euglena, if you want to look it up on YouTube. Okay, crazy what kind of stuff you can find on YouTube. But there you have it. Okay, any questions about any of these before we move on? Okay, I just want to spend like a quick moment here with a little bit of space here. Let's talk about the cytoskeleton just briefly. Cytoskeleton. Okay, just a quick summary. Okay, so there are like cytoskeleton in general either provides support, okay, for cell shape, right, for structures, or they help move things around. Okay, so there are three types of cytoskeleton that we should know. Okay, the first one is microfilament. Okay, also called actin filament because it's made up of actin. Actin is a type of proteins. You have a lot of actins in your muscle, for example. Okay. So the function of microfilament is so again, they're made up of actins. So I'm not going to write that again. And they are responsible for movements of cell. Okay, we call this like cell motility. Okay, so like not necessarily the flagellum and the cilia. Cells can move without those things, right? Okay, if you think about the white blood cells I talk about, WBC, okay, what happens is if it wants to move this way, then it's actually going to extend forward, right? The cytoplasm, it's going to grab onto something, and then the rest of the body will move forward. Okay, like it's hard to show with the diagram, but like if you imagine like my hand is like it's like the cell, right? And this is the surface. It's basically going to like grab on and then move like that. Okay, so in order to move around like that, the actin filament is going to play a role in allowing that kind of motion. Okay, so that's the first function. It will find actin filament throughout the cell, the cytoplasm, and example in muscle, right? Muscle cell. Right? There are two major types of proteins in your muscle. One of them is actin. The other one is myosin, if you want to know. Okay, so another cytoskeleton. Anybody know another example of cytoskeleton that we talked about? Yep. Thank you, Adriana. Is the intermediate filament. Okay, so this one is just to provide support. Okay, and more importantly, allows you to connects with and other cell. And so we have a lot of these like tight junctions in the body, right? Where you need to seal off areas. Imagine like your intestine, right? Like this is your intestine. If you zoom in on the cells on the wall, right? These cells need to like glue to each other, right? We don't want like the stuff to seep through the gaps between the cells. So you want to glue them together. And that's what the intermediate filaments are for. And last but not least, we have the microtubules. Okay, so they're found throughout the cytoplasm. They provide guiding tracks for vesicles. Okay, I think I posted a video in the video resources that shows you a beautiful animation of a cell, and inside you will see these tracks. Okay, so there's a big microtubules, and then the vesicles actually will move along these tracks. Okay, like like there is this molecule called kinesin, and it looks like a person without a head. It actually does. And then it carries the vesicle with the proteins in it, and it will walk along the microtubules, okay, and carries the vesicles towards its destination. Okay, it literally does walk. Like you can see it under the electron microscope. Okay, that it's travel along these microtubules. In any case, another thing is the microtubules is also something that makes up the flagellum and cilia. Okay, so those are your cytoskeleton. Great. Let's do some more polls. Polls. Where is it? Is this the poll? No. Is this the poll? Yes. All right. This is a multi-select question. Let's see if you were paying attention. Okay, just gonna give a couple more seconds. Yeah, most of you got it right. It is the RNA and the protein. Very good. B and C. Let's do another one. This is a single answer question. Okay. I can see why this one is a little bit confusing, right? Like many people choose smooth ER, but the key word here is enzyme, right? So there's no enzymes in a smooth ER, right? It is the lysosome. that has the enzymes. Okay. I know I also mentioned about detoxification of drugs and alcohols in the smooth ER, but again, right, that does happen. But here the keyword it's enzymes. That's why we know it's lysosome. All right. Here is another one. Yeah, most of you got it. This is not correct. Only one nucleus per cell, and not all cells will have a cell wall. Okay, only, well, definitely animal cells don't have a cell wall, so that makes it untrue, right? This one is actually true, right? In the muscle, you have to move them a lot of contraction requires a lot of energy. So to make more energy, you would need to have more mitochondria. Okay, and smooth ER is part of the endomembrane system. So the incorrect statements are C and D. Okay, and just one more, last one. Okay, I think I'm losing some people. Only a few of you have answered, but it has been a minute, so I'm gonna stop that. And the correct answer is microtubules. Microfilaments. Okay, microfilaments are the ones that are made up of two actins, actin strand entwined together. Okay, it says in the summary that we have right here, right, made up of actin filaments. All right, so there was your polls. Let's quickly take this up. First one, directly involved in protein synthesis. That would be the ribosome. Okay, in the process of translation, right? Protein synthesis is translation. The region inside the cell, except for the nucleus, that would be the cytoplasm. The gel-like fluid, right, that exists in the cell where everything floats in. Organelle that manages or controls all cell function in eukaryotic cell, that is the nucleus. Digest excess or worn out cell parts, food particles, and invading viruses or bacteria, that would be the lysosome. Small bumps located on portions of endoplasmic reticulum, those would also be the ribosomes. Remember, it has a large subunit and a small subunit. It's made up of RNA as well as proteins. Involved in detoxifying harmful substance and digestion of fat, produces hydrogen peroxide as a byproduct. I don't know. Did I talk about this one in the lecture, peroxisome? Can someone confirm that? Did I talk about this? Yes. Okay. So this is peroxisome, and the key term here is produces hydrogen peroxide as a byproduct. Okay. Remember, a lot of the functions are overlapping, right? Like detoxifying harmful substances and digestion of fat. You will be like, isn't it lysosome? Isn't that smooth ER? Well, they kind of do that too. Okay. But here the key word is it produces H2O2 as a byproduct. Okay. Yeah, peroxisome. Produces a usable form of energy for cell, that is the mitochondria. Okay, in cellular respiration, right? Makes ATP. Sorts and modifies contents of vesicle. That would be the Golgi body, Golgi apparatus. Site where ribosomes are produced, we make them in the nucleolus, which is the structure that is inside the nucleus, right? The seed of the peach, right? As an analogy. Provide support for cell. Well, there's a bunch of them, right? But we can just call them like cytoskeleton. Okay, so specifically there is the intermediate filament, there is the microtubules, right? Small hair structures used for movement, cilia. Forms a selectively permeable barrier around the cell, that would be the cell membrane. A whip-like structure for movement, flagellum. Contains chlorophyll, that's the chloroplast. Large sac filled with water in plants, that is the central vacuole. Produces microtubule fibers needed for guiding chromosome, that is the centrioles. All right. I think these questions, 1, 2, 3, are pretty self-explanatory. I will let you work on them, and if you're not sure, you can let me know, and I'll be happy to talk to you about it over email. Let's go through these application questions. Okay. Number 1. Speculate what will happen to an animal cell if it is placed in pure water. Do you expect the outcome to be different for cell? We're actually going to learn more about this next week. But if you put an animal cell in water, and I kind of mentioned this earlier, right? If you put animal cells in water, all the water will go into the cell. Okay, so water will enter the cell. The animal cell, right? Because there's a lot of like stuff inside, right? Like solute, a lot of stuff that are dissolved in the cell, and that tends to draw water from the surroundings. So water will enter the animal cell, causing it to swell and maybe burst. Okay, and that of course is not gonna happen for the plant. Okay, because the plant cell has cell wall. It will not burst. Okay. Next one, it's quite easy. The heart muscle needs to function continuously through life. Which organelle would you expect to be most abundant in the heart cell? So I sort of mentioned this earlier in one of the poll questions, right? What organelle do you expect to be abundant in heart cells? Anybody? Your heart never stops beating. It requires a lot of energy. So you would need to have a lot of mitochondria. Okay. Now, next one is a little bit tricky. Okay. Endocrine glands produce and secrete hormones. Which organelle would you expect to be most abundant? That means you will find a lot of it in cells of an endocrine gland. Okay, so what do you guys think? What kind of organelles would you expect to be in abundance? Tyler said rough ER, Golgi bodies. Anyone else? No? Okay, well, this is a trick question. Okay, it depends. Okay, remember there are two types of hormones, right? If we are talking about protein hormones, right? Then yes, it will be rough ER. Right? You would have a lot of ribosomes, right? Golgi, right? But if we are talking about steroid hormone, right, like the ones in the ovaries, for example, or the testicles, then you would expect to have a lot of SER, right? Smooth ER. Okay, so it depends. Right? But those are your answers. All right. Okay, so I know it's time's up. If you need to go, feel free to go. I'm just gonna take the last couple minutes going through a few questions that are like answered that a lot of people got wrong on the quiz. You might or might not have these questions because, like I said, the quiz is randomized, so everybody will have slightly different questions. But it doesn't hurt to go through them, even if you didn't have these questions. So first one, which of the following is not true regarding macromolecules? Okay, so let's take a look. First one, cell use macromolecules to signal others. Okay, that is true, right? Because signaling means like hormones. So hormone is a macromolecule, you know, I could put e.g. hormones. So that is true, right? Next one, cell use certain macromolecules to store energy. That is also true. Glycogen, right, is a form of storage, right? Lipids, that's storage. So that is not the answer. An example of macromolecule is protein. That is true. Cells produce macromolecules by process of hydrolysis. Okay, hydrolysis is when you take polymers and then you break it into monomers, right? This is called hydrolysis. Okay, so that's how you break down macromolecules, not how you make macromolecules. So for that reason, D is the incorrect statement. Okay, and the next one, subunits needed to, subunits just talking about like the monomers, right? That needs to make macromolecules is result from digestion of food, right? In other words, where do we get the raw material to build all these macromolecules that we need? We need to get it from the food, right? So this is another one, and this is one of the application questions in the study guide. So let's take a look how to do it. In a certain species of animal, 33% are adenine. Okay, so if there are 33% adenine, what's another nucleotide that would also be 33? If you have 33% adenine, you will also have 33%. Yeah, exactly right. Thymine. Okay, because A pairs with T, right? Okay, so this together, if you add them up, that makes up 66%. Okay, of all your nucleotide. So if you take 100% minus 66, you will get 34%. Okay, so the remaining 34 must be C and G, right? And they must be equally equal to each other. So half of 34 is 17. So the correct answer is 17% will be cytosine. Okay, so you might want to think that through again, and you know, rewatch the recording if you need to. Okay, this is another one that was poorly done. In regard to lipids, the term unsaturated refers to. So remember, unsaturated means there is a kink in the structure. There are double bonds, right? So is it talking about the rings in the steroid? No, it is not. Is it referring to the glycerol? No, glycerol is the same for saturated and unsaturated triglyceride, right? It's the fatty acid that differs. The lack of double bond between adjacent carbons, that's not true. The unsaturated fat is the one that has the double bond. Fats such as butter and lard, they are examples of saturated fat, so that's also not true. And then the last one, the presence of double bond, right? That's the one we want to choose. Next one, the disulfur bond, right? So this one is confusing for some people. I mean, I didn't really explicitly talk about it in the tutorial, but it was mentioned in the what you might call it, the lecture notes, right? So if you take a look, this is a diagram from the lecture notes, and it talks about the tertiary structure. There are different interactions between the side chains that will stabilize the tertiary structures. And here you can see there's the disulfur bridge, which is something that will form when two sulfur links together, okay? So the correct answer here is tertiary, okay, tertiary. Just gonna take that away. Okay, last one. In human bodies, the enzyme lactase breaks down something into something. Well, ASE is the name of the enzyme, right? I believe I mentioned that in the lecture. And so lactase breaks down lactose. Okay, so what does, there are only two options that begins with lactose, either this one or this one. And lactose is a disaccharide, right, which is a combination of glucose and galactose. So if you break down lactose, you would get those back. All right. So like I said, the class average is not bad, 73. It's pretty decent, I would say. I hope you found the quiz to be reasonable and fair, and it reflects what you learned in class. And you know, if you have any comments or suggestions, please feel free to let me know. And like I mentioned earlier, right, I'm not going to be able to share with you your individual quiz because we need to maintain the integrity of the test bank. So that is why I'm doing this instead. I chose like questions that are commonly wrong amongst many students, and I take it up. Again, I know it's not ideal, but yeah, that's the way it is. I do apologize. Any suggestions about the quiz? Any concerns about the quiz? Was the time okay? You guys were able to finish on time, hopefully? Yeah. All right. Feel free to contact me if you thought of anything. I will see you guys again next Wednesday. Okay. You enjoy the rest of the week. Thanks for coming. Bye now.